In Chapter 24, we discussed the chi-squared test. This is another type of hypothesis test that is used to show two or more categorical variables have some type of association or relationship. These are the concepts that we'll discuss in Chapter 24. Two-way tables, inference for a two-way table, the chi-squared test, and using the chi-squared test. Let's start with an example. Suppose a competitive university will admit 55 new students for the upcoming semester. Of the 140 applicants, 80 are male and 60 are female. Let's investigate whether or not there is any connection between gender and whether or not a student is admitted. If we look in the table below, we see that 35 of the 80 males were admitted and 20 of the 60 females were admitted. Furthermore, by looking at each row sum, we see that 55 were admitted in total and 85 were denied. This says that 44% of the males that applied were accepted and 33% of the females that applied were accepted. Is this evidence significant enough to conclude that the admission process discriminates based on gender? We will try to answer questions like this in today's lecture. To display relationships between two categorical variables, we'll use a two-way table, like the table of admission status and gender of the previous example. Admission status was the row variable because each row in the table describes one of the possible admission decisions for an applicant. Gender was the column variable because each column describes one gender. The entries in the table are the counts of applicants in each category. We often gather data and arrange them in a two-way table to see if two categorical variables are related to each other. To investigate the sample data, we will turn them into percentages and look for an association between the row and column variables. We will then ask the following questions. Is the association in the sample evidence of an association between these variables in the entire population? Or could the sample association easily arise just from the luck of random sampling? To answer these questions, we need a significance test. Let's look at another example. Can antidepressants decrease dependence for those with chronic cocaine addiction? A 3-year study compared an antidepressant, lithium, which is a standard treatment for cocaine addiction, and a placebo. The subjects were 72 chronic users of cocaine who wanted to break their drug habit. An equal number of the subjects were randomly assigned to each treatment. Below are the counts and percentages of the subjects who succeeded in not using cocaine during the study. For example, in the antidepressant group, 14 were successful in not using cocaine, giving us a success rate of 58.3%. The sample proportion of subjects who did not use cocaine are quite different. In particular, the percentage of subjects in the antidepressant group who did not use cocaine was much higher than for the lithium or placebo group. However, are these data good enough evidence to conclude that there is a relationship between treatment and outcome in the population of all cocaine addicts? To begin outlining a significance test for such a question, we need to explicitly define our hypotheses. In general, when we aim to investigate whether or not there is a relationship or association between two variables, we will always begin with the assumption that none is present. This is our null hypothesis. For our example, our null hypothesis would look like this. There is no association between the treatment received and whether or not an addict will use cocaine. To test the null hypothesis, we will compare the observed counts in a two-way table with counts that we would expect to observe if the null hypothesis were true. For instance, if there was no association between the treatment and the drug usage, then we'd expect the same number of successes in each of the three treatment groups. After comparing the counts with the observed values, we will argue that if the expected and observed counts are different enough, then this is evidence against the null hypothesis and thus in favor of the alternative. Computing expected cell counts is easy. We simply multiply the row and column total, then divide by the table total for each individual cell. Now you try one. Is there an association between grade average and whether or not a student plays video games? 1,808 teenagers were categorized based on their average grades and whether or not they played video games. The results are summarized in the table below. For example, 736 of the students played video games and received A's and B's on average. What would the expected cell count be for this category if the null hypothesis were true? Use the formula from the previous slide. So pause the video and unpause when you're ready for the answer. To compute the expected cell count for the category played games and received A's and B's, we first compute the row and column sums in which the observed value 736 appears. The row sum is 736 plus 450 plus 193, which is 1,379. The column sum is 736 plus 205, which is 941. 1,808 teenagers in total were sampled, so this is our table total. So if we use the formula from the previous slide, we see that the expected cell count is 1,379 times 941 divided by 1,808, which is approximately equal to 717.7. In general, expected cell counts do not have to be whole numbers. To see if the data give evidence against the null hypothesis, we'll compare the counts in the two-way table with the counts we would expect to see if there really was no relationship. The significance test uses a statistic that measures how far apart the observed and expected outcomes are, called the chi-square statistic. The chi-square statistic is a measure of how far the observed counts in a two-way table are from the expected counts. The formula for the statistic is shown below. To compute the statistic, we take the difference between the observed and expected count, square this value, and then divide by the expected count. We do this for every cell in our two-way table and then add all the results. For instance, if our table was 2 by 2, then there would be 4 cells and thus 4 terms in our sum to compute x squared. Let's return to the example on the cocaine study. Below is the observed and expected counts for the 72 individuals in the study. The expected cell counts were computed using the formula row sum times column sum divided by table total using the data from the observed table. Our table has 6 cells, so to compute the chi-square statistic, we will compute a sum with 6 terms. Each term is found by taking the difference in the observed and expected count, squaring this value, and then dividing by the expected. When we add all 6 values, in this case we get a value equal to 10.5. The sampling distribution of the chi-square statistic when the null hypothesis is true is called a chi-square distribution. The chi-square distributions are a family of distributions that take only non-negative values and are skewed to the right. A specific chi-square distribution is specified by giving its degrees of freedom. To compute the degrees of freedom, we use the formula r-1 times c-1, where r is the number of rows in the two-way table and c is the number of columns. Here are some examples of chi-square distributions for different degrees of freedom. Much like with normal curves, we compute tail probabilities of these curves to compute p-values. However, it is much harder to compute p-values exactly than for normal curves. Instead, we will compare the value of our chi-squared statistic with a table value. Here's the table we will use to determine significance. Instead of computing a p-value, we will simply compare the value of x squared to one of these values. For instance, the column with heading 0.05 is the cutoff point for x squared for varying degrees of freedom. If x squared is larger than our table value, we will say that there is evidence in favor of the alternative. Otherwise, we will favor the null hypothesis. For instance, if we had a 2x2 table giving us a df value equal to 1, we would say that there is evidence to favor the alternative at a 0.05 significance level only if x squared is larger than 3.84. So let's form a conclusion with the results of the cocaine study. Recall our chi-squared statistic is equal to 10.5. We need to use table 24.1 to assess whether this result is statistically significant. The two-way table had 3 treatments and 2 outcomes, so our statistic has 2 degrees of freedom. Looking in the df equal to 2 row of table 24.1, we see that 10.5 is between the critical value for 0.01 and 0.001 significance. This says that our p-value is between 0.01 and 0.001. So our results are statistically significant at a significance level of 0.05, for instance. Thus, we would favor the alternative and conclude that there is evidence of some association between our treatment and the success rate. Let's do one final example. Is there an association between anger and heart disease? 8,474 subjects took an anger test and were given a score of low, medium, or high. The results are summarized in the table below. Notice how the percentage with heart disease increases as the anger level of the subjects increases. Is this evidence of a statistically significant relationship between anger and heart disease? We'll perform a chi-square test to decide. Here is our two-way table with the column and row sums given. Recall we will use these values to compute the expected cell counts. The table total is 8,474 as there are 8,474 subjects. Here are the null and alternative hypotheses. The null hypothesis states that there is no association or relationship between anger and heart disease. The alternative states that there is some association. Next, our two-way table has 2 rows and 3 columns, so we will use a chi-square distribution with 2 degrees of freedom. Using the formula row sum times column sum divided by table total, we compute all the expected cell counts. To find the value of the chi-squared statistic, we take the difference between the observed and expected cell count, square this difference, and divide by the expected cell count. Adding all 6 of these values gives a value of 16.083. Finally, We compare this value to those in table 24.1. In the DF equal to 2 row, 16.083 is larger than 13.82. This is the critical value for 0.001 significance. So there is certainly evidence of an association between anger and heart disease.